Recording in progress.
Goedemorgen iedereen. Good morning everybody. I switched to English right away because the whole ceremony will be in English. That's for the, although the candidate speaks fluently Dutch, I know that, but for the international surroundings she's living in these days, and the people who are here following the ceremony by live stream, it is better to switch to English, and the whole ceremony will be in that language. Yeah, we are here, and I have to open this academic session in which Mrs. Mpabanzi will defend her academic thesis in public. Thesis is written in English under the title Targeting Renal Ammoniagenesis to Prevent Hepatic Encephalopathy. Before this committee will start the discussion and the opposition, the candidate will give a short overview of the research she performed and also a summary of her most important findings and conclusions. I ask you to start this presentation right now, and I wish you all luck and lots of success and inspiration during the coming hour.
Thank you. Good morning. Prorector, member of the assessment committee, opponent, supervisors, family, friends, online viewers, ladies and gentlemen. With the permission of the prorector, I would like to first acknowledge and pay homage to a man without whom I would not be standing here today, Professor Kees de Jong. Though grateful that his passing gave me the courage to finally finish this thesis, I am truly saddened that he's not here to cheer me on. When we do get to the time of having a wine, please do drink one on him. He was a remarkable man, a talented scientist and surgeon, as well as a great supervisor. That being said, I'm given now the opportunity to summarize in 15 minutes work that I have spent for over a decade. Let's say that I accept the challenge. The title of my thesis is Targeting Renal Ammoniagenesis to Prevent Hepatic Encephalopathy. Let me try to explain what we mean in a few slides. So ammonia is the main and recurrent theme of this work. We all use the bathroom and recognize the smell of urine. That peculiar smell of urine derives from ammonia. For the avid cleaner amongst us, you might recognize ammonia as a cleaning product. The process of generating ammonia is called ammoniagenesis. This process happens in several organs throughout the body, but for the sake of time, in this presentation, allow me to mostly concentrate on the kidney, the gut, and the liver. The main ammonia-producing organ is the gut. Ammonia is thereafter cleared in the liver in a process called the urea cycle, and thereafter excreted in the urine. The liver is commonly known as a detoxifying organ. It is therefore an essential organ in cleaning of our blood. However, sometimes the liver fails to fulfill its cleaning obligation. That can be due to an infection with a virus such as hepatitis B, or to alcoholism like me and my wines, or many other causes. So what happens when the liver fails to clear the ammonia throughout the body? There is then simply an accumulation of ammonia in the blood. And much like anything in life, too much of ammonia has dire consequences for us, one of which is called hepatic encephalopathy. Let me try it here. Which means the excess of ammonia actually travels to the brain, leading to brain swelling and potentially death. Pretty serious, you would say, right? There is currently no therapy, to effectively cure or prevent hepatic encephalopathy. And most of the interventions that are being used at the moment, they target the gut ammonia production. But what if we could shift our focus to ammonia excretion instead of production? You will have to excuse the quality of this image, but they were drawn by Professor de Jong himself over 30 years ago, so I didn't have the courage to change them. So, as mentioned earlier, the kidney also produces ammonia. Under normal circumstances, 30% of its ammonia production is excreted through the urine, and the remaining 70% goes back into the circulation. In a study published in 1992, Professor de Jong, then he was still obviously a PhD student case, was able to show in a rat model that in case of liver failure, so this one wouldn't be functioning, with high level of ammonia, a condition known as hyperammonemia, the kidney switched, and instead of excreting 30% as it initially did in a normal condition, it excretes 70% of its production, and 30% goes back into the circulation. So as I said before, most therapy, this was actually the birth of the present thesis. We call this the renal ammonia switch. So as I said before, most of the therapy concentrates on ammonia production in the gut. So what if we could, and we can see here that the kidney has the capacity to increase its ammonia excretion. So what if we could focus again on ammonia excretion instead of production? So the research question that derived from these first is there a renal switch in human, or at least do we see a change in ammonia urinary excretion in liver failure? And if we do, which ammonia regulating mechanisms are responsible for the switch? And finally, which at the end of the day is the most important, can we further enhance ammonia excretion through the urine, possibly use the kidney as a therapy organ. To answer these questions, we did some both clinical and experimental research. The first research, the study was published in 2011. Yes, I know, that long ago. The study was conducted under exceptional circumstances, namely a living donor liver transplantation. This setting is unique, as most of us will never have to live even for a minute without a liver. But patient undergoing a transplantation spends hours actually without liver. So we looked at the ammonia level in the blood and in the urine before the transplantation, during the phase without liver, which is called the anhepatic phase, and after the transplantation. So what we found was, as expected, in patients that actually have liver failure, because that's the reason why you are getting a transplant, is that the level of ammonia were high. It continued to rise during the period where actually there is no liver functioning in their body. And then once the liver was transplanted or transfused, it started going down again. Interestingly, for the urinary ammonia excretion, what we saw, it was quite low before the transplantation. It rose during the anhepatic phase, but it continued to rise after the liver was transplanted, which is interesting because you would think if the liver is back in action that the kidney would also regress in terms of functioning, but it seems like the mechanism that is linking both organs take a bit more time than you would expect, or at least that we expected. So having confirmed that the ammonia excretion through the urine also increases in humans suffering from liver failure, we continued our quest for answers through experimental research. So what did we do? We used a model of, I need to just mention this. So ammonia, as you can imagine, is influenced by many factors in the body. So we try to avoid the disturbance that could be seen by the different inflammation factor, all the other factors that might be of influence for the ammonia levels in the body, and went for what we thought was a simple model of hyperammonemia, solely induced with a diet rich in amino acid. So we fed the rat, one of the group of the rats, with a diet mimicking a bleeding in the throat. This is commonly known as an upper gastrointestinal bleeding, a complication that we often see in patients with liver failure. This upper gastrointestinal bleeding is known to also increase levels of ammonia in the blood. We obviously also had a control group which had a regular diet. Just to acknowledge this young man, the model was actually derived from human studies first conducted by my supervisor, Professor Steve Nolderdaming, and also then just a student, a PhD student. So allow me for a few minutes to kind of be technical, if I may, as it might help understand a little bit the results of what we found. So ammonia, as I said, is produced in the kidney in a cold place, the proximal tubule, and it then travels down the Lis van Henle, I'm not sure how I can say this simply, and accumulate in this area that we call the medullary thick ascending limb. And once it has accumulated, it either passively or actively diffuse into what we call the collecting tubule and is excreted into the urine. Note that at this area, though it's not given on this schema, there is a transporter called the NKCC2 that is one of the main transporters of ammonia in the kidney. So now that you have understood, because I'm sure you have, renal ammonia handling in normal circumstances, let me take you briefly through the results of our study. So what we found, I hope you can read it. So the AL, that is the regular group that had the normal diet, and HD is the group that had the hyperammonemic diet, as we call it. As you can see, over the kidney, there was more flux on the hyperammonemic side of the group that had a hyperammonemic diet. And even in terms of urinary excretion, what we could see is that relatively, this is about 73% of the renal ammonia that is being excreted, but in the group with the high protein diet, we rose up to 90% on average. So that means something is happening in the kidney. I'm sure you might have forget, but I just mentioned a transporter called the NKCC2. So the NKCC2, we tried to see if actually it was enhanced, there was an overexpression, it was more active, if I may call it that way, in the group that had the hyperammonemic diet. As you can see on these slides, is the brown color actually indicate that there is more of the NKCC2 in the group that had the hyperammonemic diet versus the group that did not have that. Because I'm lucky enough to have a co-promoter who does bioinformatics, she helped me get to the next results. Using, sorry. Oh. Mayday, mayday. It's thinking. There it is. Yes. So using a computational model together with gene expression, what we're able to predict is that several amino acid pathways producing ammonia were more likely to be enhanced in the group that was fed with the diet. These data correlated with the overexpression that we saw in NKCC2. I'm sure it might not be of very much interest to all of you, but you can see that the percentage keep on going higher. Those are the pathways that are actually likely to be enhanced in our model. So now we have an idea of how the kidney is doing that, is increasing its ammonia excretion. So the last study I would like to present is actually an interventional study where we did, though time-wise it was before we did the experimental one, but I think this is a nice example on how using ammonia as excretion of ammonia can help a patient with hepatic encephalopathy. So we use a model that is known as a crossover studies. That means actually it was the same patient that were going both interventions, only with a week interval between both study. In one intervention, we mimicked a simulated upper gastrointestinal bleed, as I mentioned earlier, and then we gave them saline, and in the second intervention, again, mimicking the gastrointestinal bleed, and then we gave them glucose. Please note that we already knew that glucose has a certain influence on ammonia levels. So what did we find? We found in the group that received saline that the increase in ammonia was actually higher than in the group that received glucose. So that means that glucose had some effect in terms of the quantity of ammonia that was being produced by the body when compared to when they just had saline. And also we noticed, same story actually, that the renal ammoniagenesis was higher when they received saline instead of when they received the glucose. So that means there is a chance or an opportunity to use a nutritional strategy, in this case glucose, to actually help patients with hepatic encephalopathy or hyperammonemia. So concluding, is there a change in urinary excretion in liver failure in human? Yes, there is. Have we been able to show the switch? Unfortunately, no. Which ammonia regulating mechanisms are responsible for the switch? from what we have studied, we can say the NKCC2 transporter, but also several amino acid pathways, and in this case, probably also glucose. And can we further enhance ammonia excretion through the urine and possibly use the kidney as a therapy organ? Yes, it is definitely an avenue that deserves more attention. So as concluding remark, I would like to say the question we try to answer are clinically still very relevant, and patients with hepatic encephalopathy deserve adequate treatments and alternative bridging the period to transplantation if they are to have one. Prorector, member of the assessment committee, supervisor, opponents, family and friend, and online viewer, thank you all for coming and for your attention. I give the word back to the prorector.
And I thank you for this presentation, which was very clear, and even I, as a former vascular surgeon, could follow it, so that is a compliment to you. We now start the opposition, the questioning, the discussion, and I can say beforehand that all members of the manuscript assessment committee are present today, which is also a compliment to the supervisory team. We start the opposition by Professor Sfiri Svetlov. She is a professor in hepatic inflammation and metabolic health at this university. She was chairman of the assessment committee of the manuscript. Professor Sfiri.
Dear candidate, I have to start by complimenting you for your presentation. I think it was very clear, and also I very, very much appreciate your sense of humor and the way you appreciated the people that helped you along the way. That was beautiful. I also would like to extend my congratulations to your promoting team. It's a beautiful thesis that you wrote. Your work definitely helped us a lot in understanding the mechanisms by which the kidney help in to control ammonia levels, and as such open the door towards future treatment. And one final comment that I want to add, I really like the way you put in your introduction the development of the hypothesis. That was very interesting to read, and especially the role of Maastricht there. So I would like to ask you first just a short clinical question. You mentioned the 3 types of encephalopathy that are present there, and especially type B was not related to liver disease. That made me wonder, how is that possible, and what are the triggers behind it, and especially to what extent the kidney is involved in all the 3 types?
Highly esteemed opponent, thank you very much for your question. I think it's actually a very interesting question you're asking, because there is currently a debate a little bit on what the name and the classification of hepatic encephalopathy should be. There was an article published by Professor Jalan and Professor Rose, who are prominent names actually in the research regarding hepatic encephalopathy and liver and cirrhosis. And one of their arguments is exactly what you just said, that why are we calling a disease where there is no liver disease hepatic encephalopathy? So they are pleading actually for a new classification of the hepatic encephalopathy as we know it, because type A is acute liver failure. Indeed, you can relate it to the liver. Type B is a portosystemic shunt. There is no liver actually involved technically. So why are we still calling that hepatic encephalopathy? And then the type C, obviously, which is the one we studied most. But for the role of ammonia, I think it's still relevant, because at the end of the day, what the liver does is clearing that ammonia. And if whether you are bypassing the liver or the liver is failing, at the end of the day, the byproduct, the toxic effect of ammonia remain.
Okay, so you assume that the metabolic switch happens in all 3 types, I assume. And then I would like to move to my next question. I was wondering, because in case of a liver failure, you mentioned there are alternative organs that takes over. One of them is the brain, and the other one is the skeletal muscles. And in relation to skeletal muscles, we have sex differences between male and females. And I was wondering, to what extent is it something that is that you see that in the clinic, for example, there are differences because of this, that the men are more resilient towards it? I would expect.
Thank you, highly esteemed opponent. I'm just going to start by in the clinic. I've been away for a long time, so it wouldn't be a fair answer for me to give that in terms of example. But what I know from the literature is obviously patients with cirrhosis, they have sarcopenia. That is one of the most, well, one of the prognostic factors for those patients. Interestingly, a sarcopenia in those patients is more pronounced actually in male than in female. So you would think male are more protected with a higher muscle mass, but I'm not sure that is actually what is happening in reality.
Okay, and taking into account the time, I will just ask you last questions with the relatively short answer. I would like to know, I mean, you have beautiful data. I would like you just to describe a little bit what is the way to translation, what are the challenges and what do you need, what are the next steps to really make it into, for example, dietary intervention that are targeted specifically the kidney or...
I think the challenges in this case remain the fact that this disease has multiple facets. So the challenge would be what we try to use a model that would avoid as many disturbances as possible. But unfortunately, we didn't get to hyperammonemia as you see when the liver is failing. So what can we do to find a way to actually diminish the noise around to actually focus on the role of the kidney and ammonia? That would be, I think, the next generation challenge.
Okay, thank you very much. I'm very satisfied with your answer, and I bring the word back to the prorector.
Thank you.
Thank you, Professor Shiri. Then we go to Professor De Meijer, if everything is okay. Yeah, then you see him on the screen there, and you can see the professor there, and the microphone is under that screen there. Professor De Meijer is HPB surgeon, especially in the field of liver surgery and liver transplantation in Groningen University. Far away was one of the reasons why he's online, but there are other reasons. Professor De Meijer.
Thank you very much, Mr. Chairman. Mrs. Candidate, first of all, please accept my apologies of not being present here today. But I do like to discuss with you your thesis, which I really, really enjoyed reading. And I'm grateful for that because I learned something new and that's always fun. I would also like to congratulate you with finishing your thesis. I would like to extend my congratulations to your friends, family, and also your supervising team. But before starting with the questioning, I would like to ask one of your paranyms to read proposition number 1.
The kidney has the capacity to increase its ammonium excretion during the unhepatic phase of the living donor liver transplantation procedure.
Thank you. That's a really interesting proposition. And with this, I would like to discuss with you chapter 4, which I think was an elegant study already back in 2011, in which you investigated ammonia levels before hepatectomy, after hepatectomy, the so-called anhepatic phase, and after arterial reperfusion in living donor liver transplantation. But I know you're not a surgeon, is that correct?
It is very, very correct.
That's the question. But I was wondering if you actually joined the transplantations during the study.
Sorry? If I was?
Did you join the liver transplantation?
Oh, I'm just going to say, do you remember the PhD student time? You are in a corner waiting for the moment when you get your samples and grateful for that. So, no.
Well, I understand. But the liver transplantation procedure itself is important for the study and for the results and for all the samples that you have received. Because I assume that there was a cava preserving technique, right? So in liver transplantation, you can remove the entire cava, but for living donor liver transplantation, that is typically not the case. But then after the liver is removed and before you have to suture in the new liver, which you actually wrote in your method section, you start with the right hepatic vein first, you do need to clamp the vena cava. Okay, and that can be a partial clamp or a total clamp. Did you know what type of clamping was used? The entire cava or partially?
I must admit that I would have to look at my supervisor and ask if he does remember. Partial.
Partial. I think that is really important, because if you do a total cava clamp, you basically obstruct the blood flow of the kidneys back to the heart, and when obstructing that, you may interfere with the measurements. And did you use a veno-venous bypass during the surgery?
I have to say no.
Okay. Pretty good. What I did notice also is that there was quite a lot of red blood cell transfusion administered during the transplantation. A median of 6 packed cells, that is nowadays a lot, and even an interquartal range of between 3 and 20. And that RBC administration could be of influence. Did you investigate the association between administration of many blood products and the timing and your measurements? Did you take that into account?
Highly esteemed opponents, thank you very much for the questions. Though I haven't been able to answer any of the surgical technical questions. I think what you're saying, we did not look at those effects, but what we do know, and I think what we show in the presentation is, for example, the gastrointestinal bleeding. So an extra blood injection might actually have effect, and we know one of the breakdown products is ammonia. So it's very much possible that the fact that the patient had so many blood products during the surgery might have influenced our results, but we did not look at it in a specific way.
I think I have to agree with you. That may explain also some of the huge variation that you see. And then 2 hours after reperfusion, you collected blood samples again, and the ammonia levels were decreased to approximately 79 micromolar. And that is still 2 times the upper limit of normal, a little less than the start values. Did you think about collecting samples later on? So after recovery, 1 or 2 weeks after surgery? And what would that difference, would have made a difference?
We did not collect sample after a week of surgery, but I think what we might probably have seen, especially if it has been a successful transplant, that the level of ammonia would have probably continued to decrease. I did read this, I think it was this morning, quickly, an article where they were looking also at a level of ammonia to estimate, to quantify actually the success rate of a liver transplant, and that wasn't seen as a good marker, because it doesn't always show what the liver is doing. But if the liver has been successfully transplanted, I would expect that the ammonia level would continue to decrease a week after transplant.
Yeah, I agree. And then my final question is, I noticed that there was actually a very wide range of ammonia excretion in the urine, somewhere between 0.02 and 6 millimoles per liter in the pre-anapatic phase to 0.80 to 10 in the post-reperfusion phase. And can you explain the wide range, the huge variance in your measurements there?
Sorry, yes. The wide range, I think, is also due to the way ammonia is being measured. Ammonia is a very volatile gas that sometimes, if it's not properly collected and properly stored and measured, it can vary quite quickly between two moments. So I think that there might also be, first of all, that change in terms of how we measured the ammonia in our samples. But also liver disease, and I think that might be the biology of each of every one of us, that it is not, we do not process things as rapidly as we have a median for everyone, but I think there is also the variation between patients. But I think one of the biggest confounder might have been the way we measured or we collected the samples, how quickly they went on ice.
Thank you very much, Mrs. Candidate. I'm happy with the discussion, and I give the word back to the chairman.
Thank you, Professor De Beer. Then we continue the opposition by Dr. Echtman. He's a gastrointestinal and oncological surgeon at Amsterdam University Medical Center. Dr. Echtman.
Thank you, Mr. Chairman. Well, first I must compliment you with the beautiful book, the achievement, your team, yourself. And someone once told me that doing a PhD defense is like a marriage, like a wedding. You wear your best dress, you have a beautiful dress, you have your friends and family here. The big difference is that after a wedding, the struggle begins, but in a PhD defense, the struggle is over. And as someone also, I finished my PhD also during my working life, I can really relate to the struggle, and I must make you another really big compliment on that. So now get to the serious part. Well, first, thank you very much, because I had to dig into the Krebs cycle and urine cycle, and total refresher course for me. That was a good experience by itself. But I want to take you back a few years back in time. You're a very young doctor, fresh from med school, starting your first job as a surgical resident. And Dr. Van Dam and Professor Oldedaming just finished their extended hemihepatectomy in a perihilar cholangiocarcinoma case. Fantastic case. They're high-fiving, going home, and you're going to take care of this patient. And just before they went home, they said to you, could you please take care of this patient, because we did a vascular reconstruction, and all went well. It was beautiful. And I show you some pictures, and wow, and they go home. And then an hour later, the anesthesiologist from the department calls you, and he says, she doesn't wake up properly, and she's confused. Could you come and have a look? So what would be your first thoughts?
Esteemed opponents, my first thought would be call Stephen and Ronald.
That's very good. That's a very good first thought. That would
And then probably go to the patient. I assume that we are passing the ABC that the anesthesiologist has already done.
Exactly.
And given the context we are in, I am probably guessing you would like me to measure the ammonia levels of that patient to see if with the acute liver, there is an acute liver failure that's expressing itself in hepatic encephalopathy.
Yeah,
That
so
would be
we
my
measure this, and it's normal.
It's normal.
Yes, it's normal.
Oh.
What's happening here?
Clinical life.
No one said it would be easy today, right?
I thought the struggle was over, but apparently not.
I'll help you a little bit, because there was a little hint, and you always have to remember what surgeons say after surgery, because they already know what's going to happen. And they were very happy with the surgery, and as I said, they were high-fiving. It was beautiful surgery, but they didn't forget to tell you they did a vascular reconstruction. So, what may have happened? And that's probably also the reason why
A leakage?
The patient may have bleeding, but the anesthesiologist did ABC, so there's no hemorrhage at the moment. So the contrary to a hemorrhage would be
A clot?
yes, no blood to the liver.
Ah, yes.
Yes, so that could be a portal vein thrombosis, or maybe they forgot to fixate the liver and it twisted. So probably you would indeed call your supervisor, and he would advise you to make an ultrasound or a CT scan. And so we did, and indeed the liver was twisted. We retwisted it, and the patient, recovered beautifully. And now it's day 5. You had some good talks with the lady and the family. She was ready to go home, and we did one final laboratory check, and the bilirubin is 150. She's a little bit jaundiced. The INR is 2.4. And again, she's a bit confused. And now the ammonia is high. What should we do? Should we give her glucose?
Without further investigation, I'm not sure that would be my first thought.
Yeah. So maybe you would call one of the colleagues at the table, right? Or maybe two.
Definitely.
Exactly.
For me, I would call everyone. But
okay, but now help me out, because I loved your book and I really like the way the thoughts move around. But I need some advice, and that's going to be my final question. In patients with post-hepatectomy liver failure, like this lady, should we give them protein or not? Is protein going to be helpful or is it going to be harmful? Because I always struggle to feed or not to feed. You say to pee or not to pee. I say to feed or not to feed. What is your opinion about this?
I tend to say not to feed. That would be my, well, let's say first, given what we did, especially with the simulated bleed, and also in our model where we are feeding, you see that it looks like it stimulated the ammonia production, at least, well, in that regard, if we are looking at ammonia in those patients, which would lead to actually more complications. So my would be this time, instead of not to feed. That would be my first, as a non-clinician, thought.
I would have the same thought, but then after 3 days of not feeding and the patient not recovering from the liver failure, everyone starts asking me, why are we not feeding the patient? Because we need to recover, right? The liver has to regenerate, so you need protein to regenerate. So I think that's a clinical struggle. Yeah. Maybe there's a solution.
But if I may, just for a quick second, when you say not to feed, glucose could be an alternative, though you cannot take it for way too long, but just to bridge a certain period. Is that not something that it is done in the clinic?
So you could give like parenteral feeding and take away the protein fraction or the fat fraction, but then you end up with almost nothing.
Yeah,
and that's the hassle. Okay, thank you for your answers. Dear Chairman, thank you.
Thank you, Dr. Eerdman. We are halfway the opponents now, the list of opponents, and we have to take a look at the clock and adjust the time you take in order to get everybody give everybody a possibility to ask questions. The next opponent is Professor Hamilder. He's sitting next to me because he's today the secretary of this board, of this committee, and he's a professor in internal medicine, especially nephrology at our university. Professor Hamilder.
Dear Mrs. Candidate, I first want to express my congratulations to you and your supervisors with the completion of this thesis. And you already mentioned that it took a long global journey to reach this endpoint, and I read that you have enjoyed this journey. And I'm a nephrologist, and I think it's very encouraging to read that the liver surgeons in Maastricht already for decades judge the kidneys as very important because of the renal switch in ammoniagenesis. So first, I want to discuss with you on your cover of your thesis, because I saw you used some flowers to enlighten the liver and the kidney. Can you give some explanation for your choice of the flowers?
Highly esteemed opponent, thank you for the question. I'm looking in the room. Actually, the artist that made the cover is sitting in the audience. And I have been told, because what I said is my thesis is about the liver and the kidney. And I got this beautiful artwork for my cover. And when I did ask what it meant, I understood that the purple flower is actually called hepatica. And it is used also in traditional medicine for, well, clearing of the liver. The yellow flower is called, I've written it down, gokshura or something like that, and that is also used in traditional medicine, but then again for clearing of renal diseases. But it was not me. It was.
It's very nice to hear that you have an artist which composed this theme, and I think it's very relevant for the subject you handle. So I want to have some discussion with you again on chapter 4, because I'm very puzzled on your observation in your well-designed study that you showed that urinary ammonia excretion increased fourfold in the hepatic phase of the living donor transplantation. And this was a very rapid response you described. And I'm puzzled, why was the urinary ammonia excretion increased after reperfusion? Because you show us that the ammonia levels are already decreased. So why would the kidney still increase the urinary ammonia excretion?
Thank you for the question. I think, to be honest, we were slightly just as puzzled as you are. As I said in the presentation, you would expect that now that the liver is indeed functioning, that the kidney would also react accordingly. The question is, did we measure too quickly the urinary expression, that the kidney actually takes some time before it reacts to the new liver is a whole new, homeostasis that is setting up. So I think we might have measured too quickly, and that it probably have we done the same measurement 2 hours later, that it would have gone down. That's obviously a guess, as we did not do it, but I think we measured too quickly.
Okay, this first explanation of you is that you have to maybe prolong the time in which you collect the urine to test how high the urinary ammonia excretion is.
Yeah.
And could there be other factors which have influenced that observation? If you look at your procedure and the circumstances of the transplantation.
I'm sure there is, but I must admit I can't think of any at the moment.
Okay, you for example, you described in your introduction that hypokalemia could lead to increased urinary ammonia excretion. Did you measure potassium during the different phases of the transplantation?
To be honest, I'm sure it was recorded during surgery, but it is actually a very good point. And maybe we should look back and see what the potassium did during those different phases. I did not personally record it for this purpose of the study, but I am sure surgically, since the patient is under anesthesia, those data can be found.
Okay, thank you for your answer. And for the time being, I have a short last question, because I'm very interested in the targets of the renal ammoniagenesis in this subject. What is the most likely to be successful intervention if you look at your data for which we have to develop an intervention? Is it the NKCC2, or is it an other component of the renal tubule which we have to intervene?
Oh, we have. Thank you. We have in these studies, we looked mainly at the NKCC2, but there are, as I'm sure you know, many other transporters that also play a role. In the last 10 of 20 years, actually, there is the rhesus transporters that have also become a prominent feature in renal ammonia handling, mostly in the proximal tubule. So no, I don't think the NKCC2 is the only target therapy, because in terms of ammonia, are we targeting the production, which is also happening in the kidney? Are we targeting the accumulation, which then you look more at the transporters? Or are we looking at the excretion? So there are different targets that you could pay more attention to. And I think what we did, especially in the last study, a nutritional intervention, seem also to be something that can give positive results for patients with hyperammonemia.
Okay, thank you for your answers. I agree on your answer that we have to look further than NKCC2. And I give back the word to the Mr. Chairman.
Thank you, Professor Hamilton. Then we continue the opposition by Dr. Kramer, or in English we would probably say Dr. Kramer. He is an internist, hepatologist at our university hospital. Dr. Kramer.
Thank you, Mr. Chairman. Dear Ms. Candidate, of course, I would like to join the previous opponents with congratulating you with this highly original research. Also, of course, including your promotion team and your loved ones. I also would like to congratulate you with your versatility. If you look at the type of research you did, it's really fundamental and translational. But when I then read your resume and looking into the fact that you did schistosomiasis research in Congo, you did a master health study, and now you are medical director of a hospital, among other things, in Africa, then I think we can conclude that you possess many talents, and the good thing is to see that you also use these talents for the benefit of others, for which I would like to compliment you. I would like to start off philosophically, because in your thesis you describe that a lot of tissues can make ammonia. And there is a lot of attention in keeping the ammonia levels in check, because high levels of ammonia are toxic, among other things, for the brain. So it's very important that we keep the ammonia levels below a certain threshold. And I was a little bit surprised to read in your introduction, or in your chapter 2 on page 29, that even under normal circumstances, you showed it beautifully in the pictures of Professor de Jong, a large proportion of the ammonia that's being produced by the kidneys, roughly 25–50%, is being excreted back into the circulation. And my question is there some kind of evolutionary or physiologically advantage of, again, replenishing the circulation with such large amounts of ammonia? Why do we do that? What's the use?
Esteemed opponent, thank you very much for the compliments. I find the question indeed philosophical, as you said. It is puzzling because it looks, when you look at the data that we have at the moment, that actually the kidney is one of the organs that contributes to the ammonia levels that we have in our body. And why would the kidney indeed produce ammonia that's going to be detoxified and again be excreted into the urine? My answer is I honestly don't know. I would speculate only that there is perhaps, maybe like the appendicit, that there is something that that ammonia does that we are not fully aware of. But we also use ammonia for example, when we use it's in the muscle, it's in the kidney, it's in the liver, it's in the brain. Even if it's as a toxic product, it seems that there is something that we physiologically need from ammonia. Otherwise, it doesn't make any sense that the kidney would be contributing actually to the arterial ammonia levels at quite high level, actually.
So we can conclude there's still room for a second PhD candidate with Professor Olderdaming to study that, for instance.
If they have 15 years, yes.
Yeah. Second question, and I would like you to take you to chapter 5. And you already described the experiments you did there with a number of Child-Pugh liver cirrhosis patients. One group, or in the first experiment, the same group getting saline infusion prior to a stimulated bleed, and a week after you gave the same group of patients a glucose infusion prior to a simulated bleed. And then you show on chapter 85 that the arterial ammonia concentrations rise, but they do less so in the group getting glucose infusion. So you show some kind of protective effect of glucose infusion on ammonia levels. Of course, I would like to use that in the clinic. Unfortunately, patients don't present themselves 12 hours before a bleed, but they present with a bleed. And my question is, how can we use the knowledge that you gained with this type of research in the clinic, where patients present with a bleed, and how are we going to use that knowledge that glucose could have a beneficial effect?
Thank you for the question. I think, well, obviously, a research setting tries to also always optimize a result by pushing confounder as far as possible. Therefore, we did indeed have the patient before bleed and after, and you can measure after bleed. But I don't think the fact that the patient already has a bleed should stop you necessarily to start with this kind of intervention. If the glucose is beneficial, as it seems to show, I don't think it's too late to continue that, while you obviously also do the ligation and everything that's needed to stop the bleed. And it might reduce the increase of the ammonia levels in the blood.
So give glucose at the moment they present at the R, and then do your ligations, etc. Okay, it's not part of clinical practice now, but perhaps we should start as of tomorrow, right? So I would like to give the word back to Mr. Chairman, and thank you for your answers.
Thank you.
Thank you, Dr. Kramer. Then we continue the opposition. It will be continued by Dr. Van Dam. He's a HPB surgeon at this university. He was the only one not member of the manuscript assessment committee, so he had to read quick, because you received the book probably as late as I received it as well. Dr. Van Dam.
Well, dear promovenda, I skip all the compliments. I'm very happy to see that you brought long ago the spirit from Africa to the Netherlands, and also the color. And I'm also happy to see that you work in a country where the colors come from the Netherlands, and the scientific spirit you bring there from Europe. So I think you are the example of our multicultural success story. So I would like to ask Dr. van Mierlo to read statement 10. And after that, I will not ask you to tell me statement 12, but I will tell it to you.
Choose a job you love and you will never have to work a day in your life. Confucius.
That was 11, but
Sorry.
But
my notes. Okay, 10.
The support team is even more nervous than the candidate herself.
Yeah, probably. You might never know what the results of your actions are, but there will be no result if you do nothing.
So doing nothing is no option. So and then statement 12, that was the statement of Professor de Jong, we all know very well. And that sounded we are capable of doing everything. We only have to show it. And I think you are the perfect example of showing it. And you also show an enormous amount of ambition, stating that we should tear down the silos between scientists, doctors and industry to make translational and clinical research possible in Africa. So please explain me how you are going to do that if you tear down the silos. How are you going to organize research in Africa?
Esteemed opponent, thank you very much for the compliments. Yes, that is the ambition, actually. I think the story behind the compliment that I got from the opponent, the esteemed opponent, about me being versatile, at the end of the day, for me, it has always been to be able to actually do something, mostly in Africa, where I come from, obviously. And the key of that is, throughout my career, I've been privileged enough to do some research. I've done some clinical work. I'm doing now some consultancy work. But at the end of the day, I think the best way to help people is by giving them the tools they need for development. And thereby, I mean education, I mean research. And a lot of research, well, life being life, has been mostly done on the European continent, or at least on this side of the world. There are a lot of things that are different there and that we do not look at. We always translate what has been done, well, with all the due respect, in male white men, and that is what we transfer into 56-year-olds Ghanaian woman, which for me, it doesn't match. So it is obviously that we need to break those barriers and move to the next step, whereby we include actually everyone and accept that we are different, because that's our strength. For me, that's not something that should divide us. It should just bring us together.
Okay, so I understand we can bring each other together with all our different capabilities and drives. But still, I wonder if you have been breaking down the silos. Should there be a dominant role for one of the players in these research programs? That can be the industry, that can be the scientist, the doctor.
Thank you. I think the answer to that is we're on our way. I work for a company. My bosses are actually in the room, and what we do is we develop and build and run hospitals. But when you do that, you have to bring everyone together. You start from you bring the architect, the engineer, but also the pharmaceutical have to come, because you need to do your pharmacy, you need to do a research department, you need to do equipment, which is again someone who's just trying to sell things and make money. So that is within the field we are operating in. We have to break those barriers, and that is, I think, a tiny but a contribution nonetheless that we are trying to bring.
Okay, thank you. I continue a bit more, because I really hope that you will use the position you have to really accelerate clinical research in Africa. So after this thesis, if you were being asked to design a clinical trial around this topic, so what intervention would you want to study?
About my thesis?
Yeah, about actually what you want is eliminating ammonia in the hyperammonia. So what would you do? What trial would you organize?
I think I would start by doing pretty much the same as what we have done here, because most of the liver disease, when you look at the African context, is mostly based around the hepatitis virus, which you don't really have as much here.
You may very briefly conclude your answer, or finalize your answer, if you wish.
Yes, please. Thank you. As I said, I think I would start by redoing what we have done there to see if the different, because I'm advocating obviously to change the population we research, but I think that would be for me the first research to see if I get the same result, and if I don't, what would be the reason of the disparity in those results. That is, I think, my first step.
Thank you.
Thank you.
Okay, it's clear that the time for the answering the questions and for your defense has passed. We now will withdraw, the committee will withdraw, and discuss the quality of your booklet and the quality of your defense. And I ask you and your company to remain in this hall until we return with the answer.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 7 miles in my rearview mirror, I know what it felt like. My goals only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror, I know what it felt like. My goals only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose that branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Get the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. To the south side, 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and that. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and that. What's on the line? I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
Dear Ms. Mpabanzi, this committee has discussed the quality of your thesis and the quality of your defense, and based on its positive verdict on both and in view of your previous examinations and qualifications, it has decided to grant you the doctor's title. The Professor Olderdamik is authorized to confer upon you this academic distinction, and the floor is to your promoter.
Please all rise.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
I promise.
By the authority vested in us by law, and in conformity with the decision of the committee here present, I hereby confer upon you, Izabiliza Liliane Mpabanzi, the degree of doctor, and grant you all the rights attached by custom and law. As evidence of this, I now present you with the degree and certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university. Dear doctor, as everything is unusual, I will start with giving you the present instead of the laudatio first.
Okay. Oh.
So what is it?
I don't know.
If I may. So this is a liver, and it's a bronze liver, and it's used by the Etruscs, also by the Greek, but with the Etrusc, the Haripecs, I think they were called. And what they did with the liver of a sheep or a goat, they looked into the future. And I think that they still look today about what the future is bringing to you, because you need decades to see your future, because what Liliane is doing, she meanders from, for all the different areas that you could do. You are so eager in everything that challenges you, that you will probably, continue with searching new avenues for the rest of your life. But this is a very special one. It's a bronze one, and it's a replica, and the replica is made by Kees de Jong. And it's given to all the PhD students. Oh. And you're the last, and I was the first. So this is mine to you. And now the circle is round. And there will no renal ammoniagenesis study being produced in Maastricht, because that was long ago. So it's something that they used to look to a forecast about what's going to happen. Most of the time, so they defined it with different fields. So about, I think, 43 or 46 different spots on the liver they used to predict the future. And the lobus caudatus, so the caudate lobe, was probably the most important. So the prominent lobus caudatus, and I think that the artist tried to depict it here, tells you a little bit about the future. And who could predict your future when you entered my room, our room, 2008 to be a DESP student, your scientific elective, a job interview of 30 seconds, 1 minute.
22 seconds.
22 seconds. Because, if she enters the room, you need to hire this. So I'm very jealous on Seert and that you're not going to call me boss anymore. Because that is gone, that passed by just giving you this diploma. So that is past history. But from 2008, 2009, you wrote the mosaic, you wrote it with a deadline, the 8th of January. So it's exactly in this time period. I think we spent Christmas time writing it. You got the 200,000 in July 2009. Yeah. Very supported by Martin Paul, the president of the university, who really pushed you and helped you, financially to go to London. Don't stay here. And I need to admit, each time that she visits Maastricht, the first thing that she does is looks at me and says, "What the F are you doing in this town, Stevie? You need to get the F out of here. It's Maastricht. We need to go to whatever." So then she had this journey and being extremely busy, prolonging all the things that you wanted. I looked at all the at the moments that you had a review of your project, the progress of your project. She did a lot of work that is not even in a book. So she did a lot of animal experiments, very difficult animal experiments with Deepak Dar and Yalda. She actually wanted to study the new potential targets, the rhesus targets, as was asked by Professor Hemmelder. We went to Zurich to visit one of the few departments of physiology, because there's almost no department of physiology anymore, with Wagner, so you really dived into it deep. And she was really busy. That's the reason it took 14 years or 15 years. And one of the most beautiful emails I could retrieve is a one from 2018. Because we waited till we had the final chapter finished. There was also a mail by 2016 in which already the valorization chapter was written, which is the last chapter of the thesis. So in 2016, everything was already finished. But we needed to publish the difficult one. So in 2018, she's working really hard. She said, "Oh, Steve, how is it? Maria, oh yeah, she's probably the first and the only person that recognized a feminine part in me, because she addresses with Maria. She always addresses me with Maria, which is my Christian name. Stefanus with brothers Maria. So thank you for being the only person that's, recognize I have a little bit of feminine touch. Anyway, 2018. Hey, Maria, how is it? I had several contacts with Sita about the paper, but she fell, she had back pain. Is there something I can do? Maybe we can have a call, and then you can, tell me a little bit about the predictions that you estimate of the fluxomics paper. And I had no clue what Sita was doing, so I couldn't. You only need to have an idea so I can finish up. And of course, I'm always available for you. How is it? Are you in Boston already? Or have you been? And I'm very busy. Double punt. Jet setting like crazy. Since January, I was in Dublin, Ardennes, Ibiza, Mali, Senegal, Ibiza, and Milan. Anyway, I'm back on the single market. And of course, I'm really enjoying life. So a good thing is that I heard that there is somebody here in the audience, which I'm very, very, very eager to meet, that kept you now, in this cage. You need to break out. Anyway, there's too much to say. I could spend the whole day in telling you how proud I am about the development that you do, but I think every member of the assessment committee recognized it, recognized the power that you have. Your last answer was actually a kind of a laudatio for what you tried to do and did in the last 5-6 years, with the massive support of your new two bosses. So really, please be careful with this beautiful jewel. Congratulations, and I'm extremely proud.
Then it's my turn again, Dr. Mpabanzi. It is a pleasure for me to congratulate you on behalf of the board of this university. We are very grateful that you, as an alumnus of this university, because that's what you are, have decided to finalize your academic education by your doctor's defense at the same university. It is always good to have you attached to the alumni list with a doctor in front of your name, title, and we are happy that you did it at this university. I also thank Professor De Meijer and Dr. Eerdmans. Where are they there? Yeah, that they are our guests today. It's good to have these ceremonies with people from other universities to emphasize the academic atmosphere in the whole country. And it was a very great pleasure for me to have you, especially you, because as a former surgeon, it is good for me to meet my old colleagues and see some parts of the field where I was in and which is fading away now. Long ago I finished my career at this university, and especially it was also a pleasure for me to have again a hint of Africa, where I worked for some years. It was in the east and not in the west where you're working now, but it was a great pleasure, and it reminded me your presentation, the way you smiled and laughed. Yeah, it was good to be back in Africa again for some time, for me then. Yeah, it is a pleasure, and I wish everybody here and a happy Christmas and happy New Year. And then I have to come to some practical points, because we have a Zoom meeting and we want you to have a picture of everything. Is there a photographer, people who are making pictures? Yeah. What we do now, before I close the meeting, I would like to ask the people taking pictures for your remembrance of this day of the whole corona. We will stand here with your diploma and also with Professor De Meijer. Then we go in a procession to the hall. There we make our official picture on the staircases of this building. That is tradition here. And somebody who makes these pictures will probably send them to you afterwards, that you have them also. And then we go in a procession to the reception room, because you're offering us a small reception in the, let's say, refectory of this former monastery, because this is actually a church we're sitting in, and we go to the dining room of the monks who lived here in former days, a few hundred years ago. It was 200 years ago. It was a Franciscan monastery. Also there you can make a picture of you and us, and on the staircase. Who is the one who's taking the official pictures? That's okay. And then having said this, I can ask you you can leave already the room, the people, while we are making these pictures, maybe with beloved ones of you also, if they stay back, if you have nobody, and then they can go for a drink. And having said this, I close this academic session. We make a picture here. Yeah.
Recording stopped.
Supervisors, please, next to the candidate. I think you might have to go a step up, because the gentleman on the screen